would like to thank all these our wonderful teams in our laboratory, in particular Stephen Jokic and Chuan Tao, who often work late night, produce the latest results, and collaborators at Columbia, University of Wisconsin, and Mount Sinai, and the funding agencies. We all know that the first FDA-approved drug for emergency use for COVID-19 is a nuclear analog called remdesivir, which is given intravenously. So the ideal drug to contain COVID-19 is a pill. And this is a press release from the day before yesterday, August 24th, which report that several papers from the Journal of Antimicrobial Chemotherapy published by Oxford University Press suggest that sofosbuvir and daclastavir are possible successful treatments for COVID-19. And the details described in these three papers. The clinical trial, small clinical trial, was conducted in Iran with an international group of scientists. It's backtracked to the end of January and early February. So before the COVID became the pandemic status, so we jump into the projects, we identify we evaluate the viral inhibitor structures of hepatitis C virus and the coronavirus replication cycle based on similarities. So we reasoned that the FDA approved the drug for hepatitis C, so sofosbuvir and paclitaxel might inhibit this the coronavirus. So and then at the same time, Alfiti performed a computational modeling, also identified that sofosbuvir might inhibit the polymerase of the SARS-CoV-2. So once we have so this is this hypothesis, then we implement all the experiments and develop the assays to identify whether the sofosbuvir is a inhibitor of the SARS-CoV RNA polymerase. So you heard from the previous talk that the SARS coronavirus 2 is a RNA virus, and in compare with DNA, because RNA is single strand, and because the RNA has this 3' hydroxyl group, this RNA polymerase has low fidelity. Therefore, the RNA virus polymerase can tolerate a lot of these nucleotide analogs as substrates. So no, the human host will use DNA as the genome. However, the coronavirus use a positive strand RNA as the genome, not involve DNA at all, to replicate its genome, or a negative strand RNA, translate the protein, so on and so forth, to replicate the virus. So coronavirus is one of the largest RNA virus genome, roughly 30,000 base pairs. So it's unique, not only has polymerase, needs polymerase to replicate to maintain the integrity, it has a proofreader carried by a exonuclease, so that the effective inhibitors need stop the polymerase, at the same time at least has some level of resistance of the exonuclease. So our research has identified the molecules, in particular sofosbuvir, has both properties. So here is the cartoon of how the virus replicate. See the RNA virus, coronavirus, enter into the host cell, release the positive stranded genome, and then translates the protein, produce RDRP, and then the RDRP, the RNA polymerase will both, transcribe the positive strand to a negative strand, and then replicate the RNA genome and produce many copies of the R positive strand RNA genome, and then yield many copies of the virus, so on and so forth. So the inhibitors we identify ideally will stop the polymerase, also has some resistance to the exonuclease, and then it might, stop the virus eventually. So for many years, we have developed this expertise of design and synthesize nucleotide analogs as a polymerase substrate, in particular for DNA sequencing. These are very sophisticated molecules. and enable the $1,000 genome sequencing. And then it shows that with these kind of molecules, not only terminates polymerase reaction, but also can regenerate the OH group. So for sequencing, it's a lot more challenging. You have to perform 2 steps. To stop the virus, we just need permanent terminator. From a chemical point of view, synthetic point of view, it's a lot of easy problem to handle. So this is the molecular mechanism, how the RNA chain grow. You have a primer, the 3' hydroxy group at the trigger, attacks alpha phosphate in common nucleoside triphosphate, and then grow the RNA chain. For the polymer, for the coronavirus replication of this genome, it involves a large number of proteins, the polymerase, the exonuclease, cofactors, helices, so on and so forth. So the ideal inhibitor that can be developed as therapeutics for COVID-19, it terminates this polymerase reaction, but also has at least some level of resistance to the exonuclease. So otherwise, exonuclease, the proofreader removed it. So we need the molecules perform both functions. So again, I mentioned based on our analysis of the viral inhibitor structure and the replication of hepatitis C virus, which is the positive strand RNA virus, similar as coronavirus, but much smaller in terms of the genome. So then sofosbuvir might be incorporated by the SARS-2 polymerase, and then it will stop the polymerase reaction. So this is the structure of sofosbuvir. This is a remarkable molecule. It's the first drug that ever developed to eradicate the hepatitis C. It's a cure for hepatitis C. So it's a pro-drug. Once entering the cell, turns to triphosphate, and then this acted as a substrate for the RNA polymerase and incorporated to the polymerase because you have this modification here at 2' side of the fluorine at methyl group, and then this acts as a permanent terminator for the RNA polymerase reaction. So again, here is the remdesivir structure, and here is the sofosbuvir structure. So the other part is roughly the same, except here is A, here is U. And you look at here, the remdesivir has a big modified group, the cyano group. Here it has OH group. The sofosbuvir just has a fluorine group at the methyl group. In the overall modifications, it's less bulky here than the remdesivir. So that's from the molecular structure point of view, the reason this should be incorporated by the SARS coronavirus to a polymerase. Then we develop a quick assay, use a single RNA template and primer, and use multi-top mass spectrometer to detect the products. And here, so we add the active form of sofosbuvir, the triphosphate, and then we obtain the SARS polymerase. At that point, in early March, the SARS-CoV-2 polymerase is not available. It's from our collaborator, Robert Kirchdorf at University of Wisconsin. You can see the sofosbuvir incorporated to the primer, and so once the sofosbuvir triphosphate incorporate, it terminates the reaction. We also evaluate some other nucleic analogs that also incorporate and terminate the reaction. So this gives us some hope that this coronavirus polymerase can take a variety of these nucleic analogs. These are existing antiviral prodrug, the triphosphate form of the prodrugs. But then we quickly move to evaluation of the SARS-CoV-2 polymerase less than a week, and Robert Kirchdorf produced this SARS-CoV-2 polymerase. We evaluated this group of these nucleic analogs and focus on the sofosbuvir data. You can see here, it turned out that this SARS-CoV-2 polymerase can incorporate this sofosbuvir triphosphate even better. It's almost all the primers transform to product and it's terminate. So at this point, at least at the molecular level, in regard to the polymerase, have identified that this sofosbuvir triphosphate incorporated by SARS-CoV-2 polymerase. and also terminate reactions. The next, we need to say whether the sulfosylate triphosphate can have some resistance to these exonuclease activities. Before we have the data, the community thought that these kind of terminators will be immediately removed by the exonuclease, therefore you cannot have any activities on the virus. So here, we develop the assay to evaluate whether the sulfosylate terminated RNA can resist the exonuclease activity. In particular, we compare with RNA terminated by remdesivir. In a way, we first use polymerase reaction to produce 3 products, RNA products. Here is RNA products terminated by sulfosylvir. S stands for sulfosylvir. Here is regular UTP, U, and here is terminated by remdesivir. So we purify these products, and then we conducted the exonuclease assay of these RNA products to see whether they have any resistance to the exonuclease activity. So again, we use multi-top mass spectrometer to measure the results. At 0 time, you see it's intact peak, only the intact RNA. After the treatment of exonuclease for 5 minutes, you can see sulfosylvir RNA has a little bit of degradation. The regular terminated RNA has a lot more termination, and the remdesivir terminated RNA has most of the termination. So this parent peak turned very small, and then remdesivir is much, much larger. And then at 30 minutes of exonuclease treatment, the parent molecule terminated by remdesivir is completely gone, and also with U. But however, you still have a appreciable amount of this sulfosylvir terminated RNA remaining. So this is very encouraging. That means at least the sulfosylvir terminated RNA has some resistance to the exonuclease activities. So this is not a comparison of the resistance of the exonuclease. We mix the 2 groups of the RNA products, one terminated by sulfosylvir, the other terminated by U, in the equal amount. At 0 time, you can see after 15 minutes of treatment by exonuclease, the regular RNA is degraded a lot more than the sulfosylvir terminated one. Similarly, we make the remdesivir terminated RNA with sulfosylvir terminated RNA. After 5 minutes, the remdesivir terminated RNA reduced quite a bit, and this sulfosylvir reduced less. And after 30 minutes, the remdesivir terminated RNA are completely gone, and the sulfosylvir terminated RNA still remaining. The other investigators, one in Brazil and the other in UC San Diego, have determined that sulfosylvir can inhibit the SARS-CoV virus replication in the lung cell and the brain cells. And there's the group of the NS5A inhibitors is target for HCV. For example, the combination drug that were identified to have benefits for COVID-19 patients is sulfosylvir coupled with daclastavir. So there's at least 6 of these molecules, all approved by FDA. They all have similar cost structures, and that they should be quickly evaluated to see whether at the virus level they can inhibit the SARS-CoV polymerase, because in the coronavirus, there's no analogous NS5A protein, and the function that probably produced by multiple of these non-structured proteins. So these inhibitors work as follows, and in order to replicate the RNA first need to be bound by these protein cofactors. Once this inhibitor bound to it and sees the RNA released, so that's why I provide another mechanism to inhibit the RNA replication. So as of today, there are roughly 2,000 patients in Iran, Brazil, Egypt, and South Africa are undergoing this larger clinical trial for this combination drug of sofosbuvir and daclazivir. We expect by sometime in October, if the results are positive, and then this drug probably can be used to treat COVID-19 patients because these are safety drugs for hepatitis C, and it's available everywhere in CVS and Walgreens and so forth. So now, and shift gear a little bit to move beyond sofosbuvir, we also evaluated a larger group of this nucleotide analog based on this FDA-approved antiviral drugs for HIV and cytomegalovirus and so forth. You can see the reason we select these molecules are because some of these, they don't have a sugar, sort of acyclic. Some of these are dideoxy, has a double bond, so on and so forth. We want to see whether these are, their triphosphate are the substrate of the coronavirus RNA polymerase, whether they can terminate the polymerase reaction, and because of the structure difference from the regular nucleoside triphosphate, whether they have any resistance for the exonuclease activity. Should quickly show some results. It turns out when you put a 2' methyl group onto the sugar, then it's they incorporate by the polymerase, but then it stops the polymerase extension. Similarly, stauvidine also terminates the reaction. When you put the biotin on the DUTP on the base, it also terminates the reactions. So these are the results for cidofovir triphosphate. This is unique. It's basically, it incorporates not only one, but sometimes two to terminate. It's sort of a delayed termination. Has some similarity as remdesivir. This is another three FDA-approved drugs. Their triphosphate form also terminate the polymerase reaction, except this one has low activity. So now, because we have this nucleotide analog with variety of structures, we like to see what's the structure impact in the nucleus resistance activities. Here with the U, you biotin here on the base. You can see at 5 minutes, you put the biotin through a long link on the base, it has some resistance. By the longer time, it's similar as a regular U. And for the stauvidine and carbovir, this is the FDA-approved drug, Aptiva. So they have very little resistance to the nucleus activities. But again, you put a 2' methyl group on the sugar of the nucleotide, then it has some resistance. For example, here's regular C, here's a modified C. At 2 minutes exonuclease treatment, the parent compound still remaining, the parent RNA. At 30-minute treatment, still has a lot of the starting RNA remaining. And then you modify the 2' with 2' fluorine, and then it has similar resistance as the regular C. So now you can ask questions. So whether we're able to find a inhibitor for the exonuclease, if we're able to fish out a FDA-approved drug that inhibits the exonuclease of the SARS-CoV-2, and then it can be added onto the drug combinations. It turns out that recently a group at Oxford University, Bandok et al., so the screen of the exonuclease inhibitors and discovered that rentecavir, which is HIV integrase inhibitor, that has some level of resistance to the exonuclease of the coronavirus. So thus, we will have a lot of combinations of the drugs. They're all FDA-approved drugs that can be tested quickly, first for inhibition of the coronavirus, and then the one has highest potency can be moved to clinical trial. For example, the combination drug can be one group.
2-minute warning if you can start to wrap up, please.
Okay. Polymerase inhibitors, the other exonuclease inhibitors, and the other group are these hepatitis C inhibitor. There's a wide variety of them, all FDA approved. So that's, I think, with this molecular engineering, structural biology, and virology, and clinical science, hopefully we can rapidly develop the therapy and contain the virus and get back to our normal life. Thank you very much for your attention.
Thank you for a huge amount of work and very exciting. Let's just hope that the FDA doesn't just announce something right before the election without testing it. We're open for questions now for Jingyue. So can you summarize and tell us what you think is the furthest ahead of all these compounds, the FDA approved drugs? Which one is the closest to being tested in patients?
So sofosbuvir in combination with danaclysivir, a small clinical trial has completed in Iran, and then a very larger scale clinical trial underway in Egypt, Iran, Brazil, and South Africa. And also in South Africa, the combination drug of sofosbuvir and daclizivir and the clinical trial for prevention and prophylactic.
And do you know what the timeline is for these clinical trials?
Yeah, by October, they expect to know the results. If the results are positive, we'll validate the early small scale trial, and then they might be available for patient use.
This may not be a question in your area of expertise, but do you know whether the FDA would accept data from Iran or South Africa or Brazil for use in the United States?
I have no clue, because it's, again, contain the virus international efforts. I think the U.S. has paid, NIH, the FDA, because it's press release everywhere, they know the results. The NIH and they follow our work. We all published. Mm-hmm. I think there's a lot of discussion also with the WHO to rapidly evaluate this, because there are syphilis drugs, in the market. There's practically unlimited availability. Right. Remdesivir is very limited. Mm-hmm.